Good morning, student. Children, we have to start with the second chapter in class 11th, subject biology. First chapter, student, you know very well, that was living world. Now this is the second chapter called biological classification. Student, in this chapter we are going to classify certain groups. Like first one will be Monera, then Protista, then fungi, then Plantae and Animalia. These are the 5 groups which we are going to study in biological classification. So we are starting with Monera, isn't it? First one is what, student? Monera. And in Monera we have the best example, prokaryotes, bacteria. So in your course the first part is the structure of prokaryotes and then its shape, nutrition and respiration. So student, just focus on this diagram. We will study the structure of bacterial cell in detail. Monera's example is prokaryote or bacteria. You see, student, this the liquid portion inside the cell. In Monera we have to study about its cellularity. It is unicellular, mostly unicellular. Then cell wall is made up of peptidoglycan and so on. So first labeling is cytoplasm. Then cytoplasm is surrounded with, you see, student, this pink color. This is the cell membrane of bacteria or prokaryotes. Then next to it is your cell wall, mostly made up of peptidoglycan. And student, one more important thing I want to tell you, that this cell membrane invaginate, like this invagination is there to form mesosomes. Student, this mesosome helps in the asexual reproduction of bacteria. When the cell divides, this mesosome, it gets deeper and deeper and the parent cell divides into 2 daughter cells. Then this is cell wall, green color, and this is, student, this one is your slime layer or film layer. These are the ribosomes. And student, very important, this area, this shaded area in some bacteria which are capsulated, this forms capsule wall. This shaded area, capsule wall, and this green structure is locomotory organ called flagellum. Locomotory organ called flagellum, and this naked nuclear material not bounded with any nuclear membrane because it is prokaryote, so it doesn't have 2 nucleus. So this structure is called nucleoid. Very important, student, nucleoid, isn't it? And these projections which helps in sexual reproduction are called sex pili. Plural is pili and singular is pilus, isn't it? And student, this extra chromosomal circular DNA is plasmid. So all these are the main labelings of bacteria. Student, let us recap once. Cytoplasm surrounded by cell membrane. Cell membrane invaginate to form mesosome. This extra chromosomal circular DNA is called plasmid. Then this is cell wall. In some bacteria, the cell wall is bordered by capsule. That is capsule wall made up of amino acid and polysaccharide. A and P we will underline. A for amino acid plus polysaccharide, isn't it? And this locomotory organ flagellum, and this is nucleoid, the genetic or nuclear material, but it is not bounded by any membrane. And these projections helps in the sexual reproduction, sex pili. Now student, we have to study Monera's shape, kind of mode of nutrition and respiration. Student, shape of bacteria, popular shape is bacillus. Bacillus means rod shape, they are rod shaped, and they are in two categories. Diplom is double, streptom is in chain. Then coccus. Coccus means student spherical, and vibrio means comma shaped. Vibrio means student comma shaped. Now on the basis of the synthesis, there are three kinds of nutrition in bacteria: photoautotrophic, student chemosynthetic, and heterotrophic. Photoautotrophic, as the name suggests, auto means self, and heterotrophic means it depends on the other organisms. Then respiration, students. Respiration are of two types. They are either strict aerobes, aerobic bacteria, or strict anaerobe. Strict anaerobe. Student, sometimes some Monera or bacteria have both the features, then they are called facultative. Facultative type, because they exhibit both the characters, strict aerobes as well as anaerobe. They can live in both the environment, isn't it? So this is all about the structure and more types of shape, nutrition and respiration. After this, we are going to study two types of the bacteria discovered by Christian Gram, that is gram positive and gram negative, just after this.